We eat very few plant species, but there's many more that we could be consuming. We've been working on underutilized crops. We're interested in developing genomic tools for these species that are very important locally and may have really great climate resiliency traits, but for example, have lower yield. We're trying to come up with ways to develop clonal seeds, meaning seeds that only have a maternal contribution and not a paternal contribution. This is potentially very interesting because it would allow you to transmit a genotype of interest, and that might be a very beneficial genotype from one generation to the next without alteration caused by sexual reproduction. The endosperm is this part of the seed that supports the embryo during its growth and development and the seedling as it germinates. When you think of a seed, it's typically the majority of the seed. It's most of what we eat when we eat a grain like corn or rice. We're trying to develop clonal endosperm, which is really challenging because of things like imprinting. We know that there are some genes that are only expressed from the paternal genome, and now we're trying to make seeds that don't have any paternal genome. One approach we're taking is to try and modulate the epigenome such that we can allow an unfertilized gamete to develop into an endosperm. In general, you can think of epigenetics as content in the genome that is not genetically encoded. So it's information that can be inherited from one generation to the next in some cases, particularly for things like DNA methylation, but is not part of the ATCG of the DNA itself. We know during the plant life cycle that there's many key developmental transition points that are controlled epigenetically or have some aspect of epigenetic regulation involved. We think that by understanding epigenetic changes that happen during normal reproduction, if we can then stimulate those in the absence of fertilization, we might be able to develop an endosperm that doesn't have any paternal contribution. We need to develop these genotypes that will be resistant to these increased stresses. The approaches we're developing are agnostic as to species, and so hopefully could be applied to any species that you can grow. If we and others can engineer these different components of clonal seed production, then that's sort of one tool in being able to sustain agriculture in the face of challenging climatic conditions.
So I'm really interested in the question of how to deliver large molecules into cells. So small molecules like maybe aspirin or ibuprofen, can kind of readily diffuse through cell membranes and get inside cells. But larger molecules like proteins or RNAs have a really hard time doing that. And we're really interested in these large molecules because many of them can be used for therapeutic purposes, especially recent advances in genome editing, where you can use CRISPR-based systems to directly change the DNA sequence inside cells, which can have therapeutic effects like correcting disease-causing mutations. Viruses are an example in nature of a system that evolved to be able to very potently infect and enter cells. And so viruses are essentially one of nature's delivery vehicles. And so in my PhD, we harnessed the virus's great ability to get into cells, but ablated its ability to cause infection. But viruses in general are foreign pathogens that might generate immune responses when delivered into human patients eventually. Another example is extracellular vesicles. And while viruses are these foreign pathogens that infect cells, extracellular vesicles are actually much more common in natural cells. They're little spherical bits of membranes that pinch off of cell membranes, and then when they're outside the cell, they exist as a semi-spherical membrane that might contain some interesting cargo, and then those vesicles can be transferred between cells. It's one form of kind of natural protein delivery that happens between cells in your body all the time. I'm really interested in studying the processes that are involved in vesicle secretion, getting a better molecular understanding, hopefully. When it comes to genetic diseases, there have been a lot of really powerful technologies developed recently for converting disease-causing mutations in genomic DNA back to like a normal DNA copy. Pairing those tools with appropriate delivery vehicles to really correct the disease-causing mutation in cells within the body, I think has enormous potential to benefit patients. And so that's one of the areas where I really see these technologies being used.
CRISPR is this great tool for, in a programmed way, precisely cutting DNA. Over a decade ago, my lab took CRISPR-Cas9, it's these programmable scissors, and basically broke the scissors. What that gave is a programmable DNA binding protein that we could deliver to any place in the genome. We then, more recently, developed an approach we called CRISPR-off that uses our dead Cas9, the broken scissors, to deliver something called a DNA methyltransferase, an enzyme that puts little methyl marks on the DNA. And these methyl marks are a signal that the cell uses to permanently turn off a gene. This seemed like the perfect approach for treating and preventing prion diseases. Prions were discovered as a new type of infectious agents that's actually fascinated the biomedical world for decades. We all express this normal prion protein. We actually don't need it. If you stop expression of the normal protein, we know that this will really prevent the infection. That then becomes this great engineering problem. How do we stop the neurons in our brain from expressing the prion protein? Here's where CRISPR came in. This seemed like the perfect approach for treating and preventing the prion diseases because we could use our programmable dead CRISPR to put in these methylation sites and to permanently turn off the prion protein. By far, the best way to deliver things to neurons was using engineered viruses, something called AAV. And the problem is CRISPR-Cas9 was just way too big to fit into these engineered viruses. Now came the final challenge, which was when we put our DNA methylases onto zinc fingers, they were not well tolerated. They had a toxicity. So we developed then a new approach, which we called CHARM. The thing that CHARM did was it didn't make new methyltransferases. Instead, it grabbed a naturally expressed methyltransferase. CHARM allows you in a precise way to just turn off any gene that's contributing to the pathology of a disease. This should be a one and done treatment. Once you've methylated the prion gene, it should never get expressed again.